So, do you have to know all the statistical models and understand every step for all of these tests? I would say it's good to understand the test that you choose to run. You should read up on and understand what it is it does so that you can interpret the results. Also, most of these tests give very similar results when it comes to the top genes, but the p-values might differ exactly how they calculate it. So when you do cutoff of a specific p-value, you might get longer or shorter gene lists depending on what test you run. And also keep in mind that fold changes can't be interpreted exactly in the same way as they are for bulk data due to all of these zeros if you just compare the means. So really understand the data, visualize the genes you get, and validate results. So really looking, I really like the violin plots because there you can really see the distributions of the gene expression between clusters and understand. In some cases, here it's clearly a shift in expression level, while here it's more a shift that this population has slightly more cells that express a gene. In this case, all cells in this population express the gene, but only about half in this population. So that it will also probably come up as differential expressed. So by looking at these kind of plots, you can start understanding what these differences mean.
Can I ask about the tests? So you said that in Seurat, it's the t-test and the Wilcox that are default.
Wilcoxon is default in Seurat.
Oh, okay.
And t-test is default in Scran and ScanPy.
And so when you use those, like, do they check the assumptions at all? Or is it because there are so many, like, do they assume that there are enough cells that you don't need to check the assumptions? Or
they just assume that you made a
okay.
decision. They don't check. I mean, they just run those tests and give the stats. So Wilcoxon is just rank based, so it sort of doesn't really consider what distributions you have.
No, I was more wondering about the t-test since that assumes.
Yeah, I don't really know. It still, I mean, it still gives reasonable results, but it assumes normal distributions, and that's not the case at all for this kind of data. So I'm not sure why that's the default, really. It's simple and works fairly well still.
Yeah.
May I also ask a question?
Yes, of course.
Just in general, the way that you now kind of describe the differential expression analysis is just between clusters. But if you would add an extra dimension, for example, control versus patients, how would you then go?
I will come to that.
Okay.
Talk about it towards the end. Because that's an additional complication.